All right, thank you so much, PJ, for the introduction. So, as PJ stated, I am a clinician researcher, and so I'm going to be shifting gears a little bit and talking a little bit of clinical medicine. But overall, we wanted to talk today, David and I, about the role of XIST-Associated autoantibodies in sex-biased autoimmunity. So just to kind of go back a little bit and talk about the disease that I really focus my research and clinical efforts on, systemic sclerosis, as PJ mentioned. I want to go over some of the clinical features briefly, and then also just review the autoantibody subsets in this patient population. Talk about some of the sex-biased differences in clinical presentation and outcomes that we see in systemic sclerosis. And then finally end with talking about this novel anti-XIST antibody that we discovered in systemic sclerosis. So just to remind you, systemic sclerosis is a disease that is characterized by fibrosis or sclerosis of the skin as well as the internal organs. And there are two major subsets that are defined by the extent of cutaneous tightening. So you can see in this figure, the areas in purple are areas that can be affected by cutaneous sclerosis. And about a third of patients are affected by diffuse cutaneous systemic sclerosis, which is thought to be a more severe phenotype. And as shown in the diagram, can involve skin tightening of the entire body. The limited cutaneous subset is actually a more prominent phenotype, and about 2/3 of patients present with limited skin involvement. And so skin tightening is limited to areas proximal to the elbows and knees, as well as the face, so sparing the truncal areas and proximal extremities. Diffuse patients, as I mentioned, we think of as having more severe disease. They usually have a rapid onset of their skin tightening around the time of onset of Raynaud's phenomenon, and they can develop severe contractures and trouble with mobility, as well as painful tendon friction rubs. The limited cutaneous group tends to have a more vascular phenotype. They have a long-standing Raynaud's phenomenon before they develop other symptoms of systemic sclerosis, but they suffer from digital ulcers, calcinosis, and can also develop internal organ vascular problems like pulmonary arterial hypertension. They too can develop problems with interstitial lung disease, joint and muscle inflammation like diffuse patients as well. But these cutaneous subsets tend to follow different natural history of disease. So you can see in the top line, patients with diffuse cutaneous disease tend to have a rapid onset of their skin tightening. Skin thickness is on the y-axis and disease duration on the x-axis. So within the first 3 to 4 years, patients develop very rapid tightening of their skin, and at the same time are at the highest risk for internal organ involvement, including interstitial lung disease, heart involvement, and scleroderma renal crisis. Over time, their skin actually softens somewhat, and their disease actually can even calm down in terms of the internal organ involvement. For limited patients, their skin doesn't increase in terms of tightening very quickly. It happens slowly over the first 5 to 7 years and then sort of stabilizes out. But they're actually at higher risk for internal organ involvement later in disease, and in particular, pulmonary hypertension. So this is a Venn diagram just to remind you of the different autoantibodies that we see in systemic sclerosis. In the blue boxes, those are the most common autoantibodies we see in association with SSC and account for about 80% of the autoantibodies in systemic sclerosis. Those are the ones that are part of the classification criteria. So the SCL70 antibody is typically seen in patients more commonly with diffuse skin involvement, but can be seen in some limited patients. And there's a high association with interstitial lung disease and digital ulcers. RNA polymerase III is associated with scleroderma renal crisis as well as cancer around the time of onset of the scleroderma. And then the anti-centromere antibody is highly associated with pulmonary arterial hypertension and most commonly seen in those with limited disease. Also, there is an association with digital ulcers and digital ischemia. So although we know that females are much more commonly affected by systemic sclerosis than males, it turns out that males actually have a more severe clinical phenotype. So this slide is from a large registry in Italy called the Spring Registry, including about 2,300 patients with systemic sclerosis. And the female to male ratio was 8.2 to 1 in this cohort. So you can see in the blue bars is males, and in the pink bars, females. And what they found was that patients who were male were much more likely to have diffuse cutaneous disease and more likely to have digital ulcers and severe internal organ involvement, including interstitial lung disease, cardiac involvement, and renal crisis. In contrast, the patients who were female were more likely to have the presence of autoantibodies, including the anti-LA antibody, the anti-centromere antibody, anti-ENA antibodies, and even ANAs were much more common in females. Interestingly, I just wanted to point out, since I'll be talking about digital ischemia and digital gangrene, this is a really rare complication and was actually found in a similar proportion of females and males, only about 1% of each. So the next study I wanted to talk about highlights the differences in clinical outcomes and survival in males versus females in systemic sclerosis. So this was a post hoc analysis of the scleroderma lung studies 1 and 2, which looked at cyclophosphamide and mycophenolate mofetil for the treatment of systemic sclerosis-associated interstitial lung disease. The SLS-1 study included 158 patients, 111 were women and 47 were men, randomized to receive cyclophosphamide or placebo for 1 year, and then followed for 1 more year for outcomes. The SLS-2 study included 142 patients, 105 were women and 37 men, randomized to cyclophosphamide or mycophenolate for the first year, and then followed for another 12 months. So the primary outcome was to look at the course of the percent predicted forced vital capacity over 24 months, and the secondary outcome was survival. So this figure shows the course of the percent predicted FVC, actually just from the scleroderma lung study 2. In the top panel, or the A panel, is patients treated with cyclophosphamide, and in the bottom panel, the patients were treated with mycophenolate mofetil. The females are shown in red and the males in blue, and as you can see clearly, the females had higher FVC, at least in the cyclophosphamide group at baseline, but improved over time compared to the males, who actually worsened initially and then improved later in disease, but not as much as the females. In the MMF group, you can see the males really didn't improve at all in terms of their forced vital capacity, whereas the females improved significantly over time. And then finally, regarding survival, the top panel shows the survival from the SLS-1 study, and the bottom panel shows the survival from SLS-2. And in both studies, the males had poorer survival over a long-term follow-up compared to females, but it was only statistically significant in the SLS-2 study. So we wondered, do anti-XIST autoantibodies play a role in these sex-biased differences in patients with systemic sclerosis? And just to remind you what Jinwoo just showed, we overlaid patient autoantibody profiles from lupus patients, scleroderma patients, and dermatomyositis patients, as well as controls, onto a structural map of XIST binding proteins, and found that the immunogenicity wasn't evenly distributed, but was really clustered around these hotspots that Jinwoo mentioned. So shown here is SPEN at the top, which was a large protein that was one of the hotspots with the highest frequency of reactivity to the sera of patients with autoimmune diseases. So we really wanted to focus on the SPEN protein. And then, as Jinwoo briefly mentioned, we took a group of patients from our scleroderma cohort at Stanford, 24 patients, sera, and we looked for anti-SPEN autoantibodies in these sera. And we found that of those 24 patients, 10 of them had high anti-SPEN reactivity, and all of those patients were female. Oops. And then we looked at the clinical features associated with those patients who had high anti-SPEN autoreactivity. And we found a very interesting correlation with those with digital gangrene or requiring amputation. As I mentioned, this is a very rare complication of patients with systemic sclerosis, about 1% incidence. And yet 4 out of the 10 patients who had high anti-SPEN reactivity suffered from this complication. So this was highly significant when compared to those without high anti-SPEN reactivity. We then went on to validate our findings with an independent cohort of patients, collaborating with Ami Shah at Johns Hopkins University. They provided samples from 35 patients with systemic sclerosis, and they too found a similar association. So 15 of those 35 patients had high anti-SPEN reactivity, and 13 of those 15 patients had severe digital ischemia or vasculopathy. So very high prevalence in this group of anti-SPEN patients. So I do want to give David some time to talk. So just briefly to summarize, systemic sclerosis is a disease with high female predominance, and autoantibodies are associated with particular clinical phenotypes. Males tend to have more severe clinical features and worse outcomes. And we are looking at antibodies to the XIST RNA protein complex to identify novel diagnostic and prognostic antibodies for systemic sclerosis patients. We think that anti-SPEN could be one of those antibodies and may indicate a high risk for digital vasculopathy in these patients. So I just want to thank all my collaborators in the Stanford Scleroderma Center of Excellence, especially PJ, Howard, Jin Woo, and Dave, of course. So I want to give Dave some time to speak, but also want to thank the NIH for funding our SCORE program.
Okay, hi everybody, good morning. I'm Dave Fiorentino. I'm a dermatologist actually. You might be wondering why is a dermatologist here? I'm not sure really, but hopefully it'll make sense later. So I first want to just thank the organizers as well for letting me speak, but most importantly the NIH for funding our SCORE Center. I'm really excited to be part of it and looking forward to it. As we're gearing up to do experiments with the SCORE grant, I wanted to talk about instead something a little bit different. I'm going to focus on some work we did with autoantibodies in dermatomyositis and in relation to women and cancer, because I hope and I think that some of the lessons we learned from that work may actually apply to what we want to do with the XIST project. And obviously any theory about autoimmunity and gender or sex preferences, they're not naturally mutually exclusive. There's probably multiple mechanisms. And so I thought I'd show you another potential mechanism. Okay, so that's my title. And I have some disclosures, they're not relevant. I'm going to quickly go to tell you about dermatomyositis. It's a different autoimmune disease. It's another systemic autoimmune disease that also has tissue-specific pathology. These patients can get a really bad rash, you can see on the right here, and in addition, they get muscle inflammation, so they can get weak and have trouble breathing, and have interstitial lung disease as well. It's another female predominant disease, about 3 to 1, female to male. And interestingly, it has these 2 peaks of incidence, so it's a bimodal distribution. So the kids get it, and that's called the juvenile form of dermatomyositis, and then there's another sort of peak in middle age. I keep kind of pushing that peak towards my age because I'm older than, I want to be middle-aged still, so maybe 60, something like that. So the other interesting thing about dermatomyositis is it's associated with internal malignancy, okay? And that could be any cancer, so it's not one specific malignancy. And it's, in adults, it's the leading cause of death, actually, in dermatomyositis, is the malignancy that they get. And it's only in the adult form, not in the juvenile form. But what's really interesting about this malignancy is it seems to be, they don't have a lifetime risk of increased internal malignancy. The malignancy risk seems to really focus around the time of onset of dermatomyositis. So if you see, if you plot years before or after DM onset, you can see most of the cases come in that 1-year window around when symptoms occur. Like systemic sclerosis, dermatomyositis patients also make autoantibodies against different autoantigens. These are 5 of them, the main ones. This accounts for maybe 85% of patients with dermatomyositis. Each patient generally only makes one of these autoantibodies, so they're mutually exclusive, and they basically create these phenotypic subgroups so that patients within one autoantibody group look much more like each other phenotypically than across antibody groups, which just sort of tells me that these antigen-specific immune responses, they may or may not be causative of disease, but if they're not causative, they're definitely reporting on something that's very closely associated with disease onset. So one of these autoantigens called is made by the TRIM33 gene. This is a member of the tripartite motif class of proteins. And at the TRIM33 does a number of things based on its gene structure. At the N terminus, there's a ring domain which confers ability to be a ubiquitin ligase. And then at the C terminus, there's two domains which actually allow it to function as a chromatin reader. So it can read histone modifications and recruit either protein complexes that actually make chromatin more accessible or less accessible. And these are the functions. So not surprisingly, that's going to, when chromatin is more or less accessible, that's going to affect things like transcription, DNA repair, et cetera. And that's what TRIM33 functions in it, but it also does have a role in cancer. And just the main thing you want to know is that it's associated, it's increased role. If you have these antibodies, you have an increased risk of having a cancer emerge, mostly around that 1-year time period. And with women, those are mostly hormonally driven cancers, ovarian, breast, endometrial. Despite that TRIM33 cancer association, if you look in the US, most of the patients still don't have cancer. It's an increased risk, but most of them don't have cancer. And so the question is, why is that? And one framework for this could be, you can imagine a tissue that becomes transformed into a cancer. You have an immune response to that cancer. That immune response may result somehow in autoimmune disease, for which we don't know what the exact mechanism of that would be. But what happens to the cancer? Obviously, that has 2 potential fates, right? The cancer could beat out the immune system and grow out, and that's the majority of the patients we see. So the cancer emerges right at the onset of DM. But the cancer could also be held in check by the immune system, either for a sustained period of time or be completely eliminated. And we postulated that this group may account for some of the, quote, non-cancer patients. It's just that cancers didn't emerge. And we thought we might be able to identify those by circulating autoantibodies. So this is work we did in collaboration with Olivia Casciolo-Rosen at Johns Hopkins. These are all patients with anti-TRIM33 antibodies. So it's a subset of DM that's at high risk for cancer. And we looked at the patients who did have cancer emerge and didn't have cancer emerge. And we did immunoprecipitation studies. Each lane is a patient sera immunoprecipitating protein extract. So you can see obviously all of them are making TRIM33 because that's our cohort. And the one striking thing is that you can see just from the gels is that generally patients who don't have cancer have many more antigens, have a more diverse antibody response. So we decided to go after some of those and identify the nature of those autoantigens. And one of those turned out to be a protein called CCAR1, which also has a number of roles in the cell. And I think with a lot of these RNA binding proteins that are antigens, that's the theme, is that they have lots of potential functions. And so it's a little bit difficult to try to figure out which one or ones are the relevant functions in what we're talking about related to autoimmune disease. So this mediates transcriptional activation, mostly by hormone receptors. So that's very interesting. Nuclear receptors. It's involved in other processes as well. And so we went back to the cohort and identified all the patients that make this antibody. And we actually had a big cohort at Stanford and one at Hopkins. And the basic experiment was to compare the patients with TRIM33 who did not make this antibody and those who did, and look at the SIR for cancer emergence. So you can see without antibodies, you have a high SIR for a future cancer in the next 3 years, but that is much attenuated by the presence of CCAR1 antibodies. Now, what I'm showing you here are all the cancers that developed in our study. So you can see, right, as a function of the time of DM onset. So you can see all the cancers, each dot is a patient, right? They all kind of cluster around the onset of disease, like I just mentioned. Now, the cancers that do appear in the CCAR patients, a few do seep through. But what's interesting, those are shown in blue. They all come, most of them all come up at a later time, which would be consistent with a cancer being held in check. We found another bunch of antigens and validated them in the 2 cohorts. And just to show you, this is just to show you that there's other antigens. Many of them also seem to be conferring cancer protection. And you can see some are very common, some are only found in the index patient in which we identified it. So it's almost like an immunome for their cancer. But the interesting thing is there seems to be some dose response. So if you just look at CCAR1, the patients who had a cancer emerge and didn't, the patients who have cancer emerge tended to have lower titers of CCAR1, which is also sort of interesting, right? If you think maybe the antibody itself or the immune response itself is actually having something to do with it. But beyond CCAR1, if you look at all the add-on autoantigens, you see that most of in this graph, you can see with the patients who got cancer, which is the light gray bar, most of those patients are accounted for by having no add-on autoantibodies. And then maybe the rest of them had one add-on autoantibody. But you can see the patients who did have cancer, if you have more than one, you really aren't going to have a cancer emerge. So there's some sort of weird dose response going on here. So for this one, just focus on the lower right, left. This is just showing you on the left, these are each of the different add-on autoantigens. And then we have the frequencies of them. And this is in the no cancer cohort. And what I want to just point out, there's a lot of patients who have combinations of these antigens, which I just mentioned. And it turns out many of these autoantibodies occur together way more than you would think by chance, right? So if you kind of string that up as bars and connections, you find that they actually almost form hubs of very tightly connected autoantibodies that seem to co-occur. And there really seem to be maybe 2 in our group here. There's sort of 2 hubs that we found between cancer protective autoantibodies. And so the question, I was intrigued by that. That could mean a lot of things. You can't really make much of that. You don't know what that means. But one possibility is that they're either participating in a functionally, in the same functional process or even potentially being physically related. And so if you go back and you say, okay, these antibodies were identified in this TRIM33 positive cohort, which of them co-occur and seem to either limited completely to the TRIM33 group, because remember there's the whole bit of DM patients that don't make TRIM33. So if you look and see which are enriched in the TRIM33, it's the ones with these red arrows. So we postulated that some of them may interact, and if you look, indeed CCAR1, if you do immunoprecipitation blot studies, CCAR1 does interact with TRIM33. We haven't really looked at the other antigens, but it brings up this concept that maybe these are forming a structure. Which is, you can see, is becoming the theme. Multimeric protein structures which may or may not have RNA in them. I'll talk about that in a minute. So one thing that was really striking about some of these autoantibodies is they're almost only found in women. So CCAR1 is 95% women. It's very rare to see that in men. And we know CCAR1 is associated with absence of cancer emergence. And so it is indeed true that these antibodies cause a decrease in emergence of hormonally driven women cancers. So breast cancer, ovarian, uterine cancer. Okay, so that's the first thing. Now, if you look at the juvenile population, those patients actually also make these antibodies. So these antibodies don't seem to have only something to do with cancer, which is interesting. So keep that in mind. And if you look carefully at the juvenile peak of incidence, you actually find that there is this, in the open bars are women, females, young girls, and you can see that there's a peak right here around the time of the, and I think, what's happening right there in this person's life? Well, if you're a parent of women, of girls, which I am, things can get dicey because the hormones are going, right? And they're in pre-puberty. So that's an interesting sort of observation. And so now Jinwoo was saying for people who don't like speculation, you can stop listening now. But one possibility here is that putting these facts together, okay, you have this peak in juvenile DM, right, around puberty, beginning of puberty. And then in the adult population, you have the same antibodies that seem to be protecting against hormonally driven cancers, okay? And then you also have this knowledge also that I didn't tell you that DM is also can be induced by pregnancy itself. And so this gets back to maybe there is something about the hormone here. And again, not mutually exclusive with XIST for sure. I just want to make that point. So what about that could it be? hormone receptors function as dimers, and they bind to DNA, right? This is the estrogen receptor. The ERE is the estrogen receptor DNA element. And maybe it has something to do with the process of responding to estrogen. So it turns out the estrogen receptor doesn't act alone to activate or repress transcription. It actually binds a number of either repressor complexes or activator complexes. So there's going to be a big group of proteins at this site, okay, with DNA. So you've got DNA, nucleic acid, and then a bunch of proteins. So I went back and looked at all the patients, all of the antibodies that are in a hub with TRIM33. Okay, those are shown in red on the left. And then on the right, these are all antibodies, important antibodies in DM that don't form hubs. They don't seem to occur in relationship with the TRIM33 antibodies. And virtually every single one of those is shown in bold is known to interact with the estrogen receptor. And these aren't because like someone did a pull down with an estrogen receptor and got 3,000 proteins and I found it in the list. These are individual papers where they were looking at the two together and having functional consequences and binding each other. And so is the estrogen receptor or hormone receptor complex potentially a source of autoantigens? And I think it's not so crazy to think that that could be true, actually. You know, when you have enhancers functioning to modulate transcription at promoter sites, these groups of proteins can undergo what's called liquid-liquid phase separation, and they can really form like a gel within the nucleus. So it's sort of like a membraneless compartment to concentrate these proteins and make them the process more efficient. So you can imagine that's a really potential source of the immune system not liking that and recognizing it. So I think I'm just going to finish. Hopefully I'm on time. I don't know. I think I am. Oh, I got my 2 minutes. I'm on time. All right. So in conclusion, so you have this antigen, TRIM33, that is a dermatomyositis autoantigen that's associated with cancer emergence. We don't know why. We found these add-on antibodies. Many of them are associated. They ameliorate the risk of that cancer emergence. And what's interesting about many of these antibodies, including CCAR1, is that they seem to co-occur commonly with TRIM33, okay, which may indicate physical binding. We've shown that in the case of CCAR1. And maybe what's happening is that any time in the person's life, the female life, when there are changes in estrogen response, maybe that you can imagine that complex is going to be changing in its identity, it's going to be changing in its interaction partners, and so at pre-puberty, pregnancy, or if you have a cancer that's emerging that's hormonally dependent, may be a stimulus to the immune system to cause these antibodies. So I think, and I guess that's my point, I think the idea of sort of exhausting the number of antibodies, like, you know, really trying to do a full search of antibodies may start to give you clues by who they're associating with, what process you're actually looking about, what is the structure and where is it? Because these proteins form in multiple different structures with multiple different processes in the cell. So with that, I'll just say thank you to everybody. Lori is my clinical partner, and Julian's here. He's our wonderful fellow, and Matt Lewis is a dermatologist who's helped us collect samples, and definitely the SCORE group, who I've personally been working with. And the work, really, all this autoantibody work wouldn't have been done without a collaboration with Livia Casciola-Rosa and Anthony at Johns Hopkins. And yeah, just a big thank you for the NIH as well for funding our project. Thank you.